Welcome to the Chapter 7, Part 1 lecture on passive transport. You should use the information in this lecture to complete the Chapter 7, Part 1 guided notes, which of course you should complete before you attend class. This lecture will discuss some of the various methods that cells use for exchanging molecules with their environment. To understand this, you first need to understand how the cell membrane is put together. We'll also discuss how molecules move in general in this lecture, and we'll discuss some methods of passive transport. Now you're going to find that the information in this lecture is basically the same as in the pre-lab for Lab 5. I told you that we'd go over it together before the post-lab was due, so you'll want to have that lab handy because some of the year turns refer to the pre-lab. Let's get started. The general motion of molecules is known as Brownian motion. This lecture and your pre-lab for Lab 5 will just refer to this movement as just movement by itself. Molecules are constantly in motion in every direction. Now inside a gas, those molecules will have the most kinetic energy, so they will have the most range of motion. They're moving around a lot. Inside a liquid, they have a little less kinetic energy, so they move a little less. And inside a solid, they have the least amount of kinetic energy, so they can't move very much. But even inside a solid, those molecules are going to be vibrating slightly. So remember, when you do your pre-lab and when you answer questions about movement, if it's just movement by itself, molecules are always moving in every direction as much as they can. Now the term net movement is a little bit different than just the term movement by itself. It's different than plain old Brownian motion. Net movement implies that there's some sort of an imbalance in the system, and because of this imbalance, molecules are moving in a specific direction for a specific reason. What kind of an imbalance am I talking about here? Well, here, for instance, we have an imbalance in concentration. We have a high concentration of these molecules on one side of this barrier and a low concentration on the other side. So that makes the molecules want to move from high to low. There's some difference that's making it move in a direction. When we have a difference in concentration or in pressure in two different areas, we say that a gradient exists. And when gradients exist, there will be net movement or movement in a specific direction. So if you're asked a question on your pre-lab or in class about the net movement of molecules, the answer is always going to be something like top to bottom, inside to outside, right to left, something like that, in a specific direction. In this chapter, we're mainly going to be dealing with concentration gradients and how cells use concentration to make molecules move where they want to go. There's a couple of things to remember about concentrations. First, they can be measured in percentage or molarity, and we'll talk about molarity in a moment. When you're dealing with percentages, it's important to remember that the percentage of the solute and the solvent is always going to add up to 100%. So here, for instance, if we have a solution that contains 10% salt, we know automatically that it must contain 90% water, because 90 plus 10 is 100%. The other thing to remember about percentages is the higher the percentage, the more of that component is going to be present in the solution. So for instance, a solution with 70% solute is going to have a lot more solute than something with 10%. Another way to represent concentration is, of course, good old molarity. And there's really just a couple of things you need to know about molarity for this chapter. First, you need to remember that the higher the molarity of a solution, the higher the amount of solute it has. So a 1 molar salt solution has more salt in it than a 0.5 molar solution. The other thing you need to remember is that the amount of solute and the amount of solvent within a solution are indirectly related. So the higher the amount of solute, the less water you're going to have in your solution. That means that a 1 molar salt solution will have more solute, but less water than a 0.5 molar solution. Gradients exist anytime you have two areas that differ in some characteristic. For instance, here we have a concentration gradient. We have an area that has a high amount of solute here and a low amount of solute here, so there's a difference in the solute concentration. Therefore, we would say that a concentration gradient exists between these two areas. Now we have a few vocabulary terms that you need to know that have to do with concentration gradients. If molecules are moving down or with a gradient, that means that they are moving from high to low concentration areas. So for instance, if we have a molecule moving from this side of the solution to this side, from high to low, we say it's moving down or with the gradient. On the other hand, if we have a molecule that's moving up or against the gradient, that means it's moving in the opposite direction, from low to high concentration. So for instance, if we have this little molecule over here and it's moving from an area of low concentration and getting more crowded to an area of high concentration, we would say it is moving up or against the concentration gradient. Which of these motions do molecules prefer? Well, in general, molecules like to move down or with the concentration gradient when they can. In other words, they like to move from where they're all crowded up to where they're all spread out. Think about this like getting on an elevator. If you got on an elevator and only one other person was on the elevator and they stood really, really close to you, you would feel really uncomfortable. Molecules are like that too. They like to spread themselves out. They like to have some social space. So in general, they try to move from areas where they're crowded to areas where they are not crowded. So now that you understand a little bit more about how molecules like to move, let's circle back around and apply this to living cells. How do living cells control which molecules move from the intracellular environment to extracellular and vice versa? Well, there's two major types of transport that cells like to use. The first type of transport is known as passive transport. And passive transport allows molecules to move the way that they normally would like to. They go where they want to go. Remember that molecules generally like to move from areas of high concentration to low concentration, down the concentration gradient. So here we see that. We see that these little triangles are crowded on the extracellular side, and they are less crowded inside, so they want to flow through the membrane and spread themselves out, moving from high to low concentration. Passive transport is the preferred type of transport by cells because this requires less energy. These molecules have kinetic energy, and they can use that to move where they want to go. But this type of transport requires no extra ATP energy on behalf of the cell. So because the molecules are moving the way they want to anyway, the cell doesn't have to expend any extra energy. So they prefer this method. Another method of transport is known as active transport or active processes. Active processes are the opposite of passive processes. In active processes, molecules are forced to move against their will, against or up the gradient, where they don't want to go. For instance, here we see a low concentration area of diamonds in the extracellular environment, and you can see that there's a higher concentration inside the cell. The cell is having to force this little diamond-shaped molecule through that membrane and across into the intracellular environment. It's making it more crowded, so it doesn't want to go that way. Now, because molecules are being forced to move against their will, against the gradient, the cell does have to expend some energy. And this energy is always going to be in the form of ATP. Here you can see an ATP molecule being broken down for energy to power this reaction. All right, now I told you that this lecture would help you with the pre-lab for Lab 5, and it definitely will. At this point, I would like you to get that pre-lab out, go to page 3, and work on the year turn that's at the bottom of page 3. You should now have enough information to answer it confidently. Make sure that you bring your completed pre-lab to class with you, lecture class, because we will work on it there, and you'll be able to confirm your answers. The rest of this chapter is going to focus on passive processes the cells use to move molecules where they want them to go. The first and most common type of passive process is simply called diffusion or simple diffusion. And this is the movement of molecules from areas of high concentration to low concentration, all by themselves. Here, for instance, we have a drop of red dye that's been placed into a beaker of water. Now, within that drop, those dye molecules are going to be very crowded. That represents a very high concentration of dye. So almost immediately, they are going to experience a net movement. They are going to want to move in a direction because of a concentration gradient. So they're going to want to move from in the drop to outside the drop. They're going to move into the water. And conversely, the water is going to want to diffuse into that drop because it's moving from high to low as well. We call that a net movement. Now, eventually, the dye molecules are going to get nice and spread out in that water, and they're going to reach what we call a net equilibrium, or sometimes just equilibrium. At that point, there is no more net movement because they're spread out. They don't have to move in any particular direction because they don't have to. They don't feel like it. They're happy where they are. Here we have some practice problems similar to those on Pre-Lab 5. What we have here are two solutions that have been placed in adjacent beakers. They have different concentrations of salt and water, and they have a semi-permeable membrane between them. So it's some kind of a barrier, but it's a barrier that the salt and the water can work its way across. What's going to happen to these molecules when you put them in this situation? Right? I want you to think about the movement of the salt and what it wants to do, and the net movement of salt. Remember that those two terms are different. See what you come up with. So what answers did you come up with? Let's discuss it. First, I'm asking about the movement of salt when these two solutions are first exposed to one another. Remember that anytime I ask about just plain old movement, I'm talking about Brownian motion. So the answer here is going to be both directions. Molecules are always moving in both directions a little bit, regardless of the situation. Now next, we're going to talk about net movement. Remember that net movement is in a particular direction because of some kind of gradient. Because there's a higher concentration of salt on the right-hand side than the left, the salt is going to want to move from right to left. That's its net movement. Next, let's say that these two solutions have been allowed to equilibrate with one another. They've been allowed to diffuse together however they wanted to. And now we have a situation where they are at equilibrium. What will happen to the movement and the net movement of salt? See what you come up with. So we have this solution that's at equilibrium. Now you might think, okay, well, there's no reason for the molecules to move around now, so the movement might be neither direction. But remember, anytime I'm asking about just movement, the answer is always both directions. Even though equilibrium has been reached, those molecules are still going to be moving around both directions all the time. Okay, that's just the nature of Brownian motion. Now, what about net movement? After equilibrium has been reached, there is no net movement. So the answer to the net movement question would be neither direction. There's no particular reason for a molecule to move left to right or right to left, so it's neither direction. Several different factors can affect the rate at which diffusion occurs, and the first is tonicity. Tonicity is another word to describe the strength of the concentration gradient. So the bigger the difference in concentration between two solutions, the higher the tonicity. So how does this affect diffusion? Well, here we have a situation where we have a big difference in tonicity. This side is highly concentrated. This side has much less solute in it. In this situation, the solute is going to want to move from high to low really, really fast. And conversely, the water in the solutions is going to want to move the opposite direction really, really fast because there's a big difference in their concentrations. On the right-hand side, though, diffusion is going to be much slower because there's less of a difference in tonicity. The concentration gradient isn't as strong. So the water will want to move in this direction, but slowly. the solutes will want to move in this direction, but pretty slowly. So the greater the tonicity, the higher the rate of diffusion. You can apply this concept to part 3 of your post-lab results for lab 5. Think about what you did. In part 3 of the lab, you constructed dialysis tubing cells containing different solutions with different concentrations of glucose, and you soaked those cells in water. You kept track of the change in mass before soaking and after soaking. In which of those tubes would you expect the fastest rate of diffusion? In other words, in which of those tubes would you expect the cell to gain the most weight? Is it the one that just had a little bit of glucose inside when you started, or the one that had a lot of glucose inside when you started? Think about it. Temperature is another factor that can affect the rate of diffusion, and you know this from just working in the kitchen. I mean, think about this. What's easier to work with when you're making coffee or tea or ramen noodles? Well, warm water is usually easier to work with than cold water. Things don't mix up as well in cold water. Now, why is that? Well, when molecules are warm, that means they have more energy. More energy means they can move around faster. And the faster they're moving, the more likely they are to diffuse. So warm solutions tend to diffuse faster than cold solutions. Cold solutions kind of just sit there. They don't have enough energy to move around. You can apply this concept to part 2 of your post-lab 5. In part 2, you had different agar plates, and you had methylene blue and potassium permanganate drops diffusing through those plates. One of the plates was kept in the refrigerator, and the other 2 were kept on the counter. In which of the plates would you expect to have a faster rate of diffusion or a greater rate of diffusion? The ones that were at room temperature or the cold one that was stuck in the fridge? Think about it. The size of the molecules involved can also affect the rate of diffusion. And I kind of think about sports when I think about this phenomenon. In sports, the big guys are usually the strongest guys, right? But they're not always the fastest guys. It's those little zippy guys you gotta watch. They're usually faster. And the same is true of diffusion. If you have big molecules and small molecules, the small ones usually diffuse faster than the big ones. The big ones are a little bit slowed down. It takes more kinetic energy to get those guys moving. You can apply this concept to post-lab 5. In part 2 of your lab, you were comparing the diffusion rates of 2 different kinds of molecules, methylene blue and potassium permanganate, through agar plates. Those 2 molecules differ greatly in size. So which one would you expect to diffuse faster and more in your agar plates? The really big molecule or the really small molecule? Think about it. There are several different specific types of diffusion that exist, and the first one is known as dialysis. In dialysis, we're going to focus on the movement of the solutes across a semi-permeable membrane. So like a membrane like dialysis tubing or a cell membrane. In general, molecules still like to move from areas of high concentration to low concentration. The medical procedure known as dialysis has to do with your kidneys. So what do your kidneys do? Well, really, your kidneys maintain the content of 2 different solutions inside your body, your blood and your urine. Your kidneys filter your blood something like 45 times a day, and the kidneys are the organs that get to decide what solutes and water stay in and what comes out and leaves your body as urine. When the kidneys are failing or not working properly, people have to go in and get dialysis, which is an artificial filtering of the blood. And in this filtering, certain solutes are pulled out so that the blood doesn't become toxic to the body. It's definitely a temporary measure. It's not as good as a functioning kidney, but it can keep people alive over an extended period of time. Another type of diffusion is known as osmosis. Osmosis is the movement of a solvent across a semi-permeable membrane from an area of high concentration to low concentration. Now, because we're talking about living cells and cells contain an aqueous solution, the solvent of choice here is going to be water. So osmosis is really the movement of water from high to low concentration. When we talk about osmosis, we're really talking about the movement of water between solutions across a concentration gradient. When we compare the concentrations of different solutions, there are three words we can apply to them: hypertonic, hypotonic, and isotonic. When we use the terms hypertonic, hypotonic, and isotonic, what we're really doing is comparing the concentrations of two solutions to each other. Now, when you do this, you can either look at the concentration of solute or solvent, whichever makes the most sense to you. I prefer to look at the concentrations of solutes. So let's say we have two solutions here, A and B, and we're trying to appropriately apply these terms. Solution A has more solute than B, so A is the hypertonic solution of that pair. In science, hyper means above. So for instance, if someone is breathing too fast, you might say they are hyperventilating. They're breathing too much. Concentrations that have more solute are considered hypertonic. Conversely, a solution that has less solute is going to be considered hypotonic. Hypo in science means below. So think about a hypodermic needle. Where does a hypodermic needle go when you get a shot? Well, it goes under the dermis, so it's hypodermic. If we compare solutions A and B to one another, solution B would be considered hypotonic because it has less salt, less solute than solution A. Sometimes solutions have exactly the same concentrations of solutes and solvents, and in that case we call them isotonic. We would say that solution B and solution C are isotonic to one another because they have the same levels of solute and solvent. Keep this in mind when you use these terms. When you compare the concentrations of two solutions, they're either going to have exactly the same concentrations and be isotonic to one another, or one is going to have more solute than the other. If one has more solute, it is hypertonic, and the one that has less solute would then be considered hypotonic. So they're either going to be a pair of hypertonic and hypotonic solutions, or they will both be isotonic to each other. What I'd like you to do in this your turn is to compare the concentrations of these solutions in pairs. And then I want you to fill in a series of sentences using the words hypertonic, hypotonic, and isotonic. For instance, you might compare beaker A to beaker B, and you would fill in the sentence, A is blank to B. So A is either going to be hypertonic to B, hypotonic to B, or isotonic to B. And then create sentences for the remaining beakers. Compare all of them to all of them. Bring your results to class. When we have solutions with differing concentrations of solutes and solvents, and we expose them to one another, those solutes and solvents are going to want to move, and generally they want to move from areas of high concentration to low concentration. Now, when we're talking about osmosis specifically, we're talking about the movement of water. So here's the kind of rule that describes how water will move between these solutions. In general, water wants to move from hypotonic solutions towards hypertonic solutions. So I kind of remember this. They move from O towards from O towards So let's say that you're presented with a situation like this, where you have two solutions of differing concentrations, and you're asked to predict how the water will move in the situation. Well, the first thing you want to do is identify which solution of the pair is hypertonic and which is hypotonic. In this case, solution A is hypotonic because it has more water and less solute, and solution B is the hypertonic solution. Remember that water always likes to move from O towards so you could say that water will move from A towards B from left to right, given the option. Now the salt will want to move in the opposite direction if it can, but it kind of depends on whether that solute is capable of crossing this semi-permeable membrane, and that depends on which kind of membrane you're dealing with. So what does all this stuff have to do with living cells? Well, remember that cells are both filled with solutions and live in solutions. The intracellular environment within a cell is a solution. It's mainly water, that's the solvent, and a bunch of different solutes, including salts and electrolytes and ions. The extracellular environment is also a solution, and it's going to differ in its concentration of solutes and water. Cells will make use of this concentration gradient to make molecules move where they want them to go. Now you might be asked to identify which way solutes and water will want to move when cells are placed in certain situations. How do you go about doing that? Well, first of all, you need to identify whether the intracellular solution is hypertonic or hypotonic, and the same with the extracellular environment. In this case, we have more water outside than inside, so this is going to be your hypertonic environment, and the extracellular will be the hypotonic environment. Water moves from hypo towards hyper, so in this case, water is going to try and flood through that membrane and fill up that cell. If the situation were reversed, the water would leave the cell would get dehydrated because the hypertonic solution outside would pull that water out. Let's look at some examples of cells being exposed to these different kinds of environment. First, we'll start with an isotonic environment. Red blood cells inside your plasma, that's the liquid of the blood, live in an isotonic environment. The plasma, the liquid, has about the same concentration of solutes and water as the inside of the red blood cells. The red blood cells living in this nice isotonic environment have a particular shape. They're shaped like little coins, and they have a dent in either side. That shape's called biconcave. And they're pretty happy like this. Now, what would happen to one of these poor little red blood cells if we took it out of its nice isotonic environment and threw it in a really hypertonic environment, like really, really salty water? Well, here we have the result. If you throw a red blood cell into salty water, that salty water will be hypertonic compared to the inside of the cell, which will be hypotonic. Water, because it travels from hypo towards hypertonic solutions, will leave the cell. In other words, the cell gets all dehydrated, and the cytoplasm of the cell starts to shrink up. Look at how spiky and weird this sad little red blood cell is. What you're seeing there is the hemoglobin protein inside actually kind of poking through the membrane. The cell obviously can't do its job like this. It's pretty unhappy. Now, we do have a science word for the shrinking. We can't say that it shrinks. We have to say that that cell crenates in a hypertonic environment. Next, what if we did the opposite? What if we took one of those poor little red blood cells out of its happy little isotonic environment and threw it in a very hypotonic environment, like really, really pure fresh water? What would happen? Well, that kind of an environment would be hypotonic compared to the inside of the cell, which would be hypertonic. Because water travels from hypo towards hypertonic solutions, it would flood into the cell through that membrane. And look what happens to it. Now that little cell doesn't have its happy little disc shape anymore. Now it's all swollen. And it can't do its job like that either. Sometimes when cells are exposed to hypotonic environments like this, they'll even explode. And the sciencey word for exploding is lysing. The cell will burst. So obviously these dramatic changes in shape are not good for your red blood cells. This is the reason that if you go to the hospital and you need to have blood volume replaced, like you've lost some blood and you need some more liquid in there, they don't just put distilled water in your veins. And they don't put super salty water in your veins either. They put something like Ringer's solution that's isotonic to your red blood cells to prevent them from changing shape and taking on damage. How do your red blood cells defend themselves against these kinds of changes? Well, most of the time your body is able to maintain the homeostasis of the chemical content of your blood just fine, so that your blood plasma remains isotonic to your cells and they don't change shape. There are certain severe conditions, though, that can cause your blood plasma to become hypertonic or hypotonic. For instance, if you get really dehydrated, your blood plasma might become hypertonic to your red blood cells and they'll shrink up like this, which is not good. Conversely, if you drink a lot of fresh water, and it has to be a lot, your blood can become hypotonic to your red blood cells and they'll swell up and burst, which is even worse. That can affect your kidneys. But most of the time, organs such as the kidneys and the lungs are able to keep your blood isotonic so that your red blood cells remain healthy. Now, we've gone through a lot of things in this lecture so far, but you are finally now ready to tackle that big, ugly multiple choice section in your pre-lab for Lab 5. So please go do that. Get out your pre-lab, go to pages 4 through 6, and see what you can do with it. Bring your results to class. Simple diffusion, osmosis, and dialysis work really well for moving small molecules that are not charged, are nonpolar, across that membrane. And these processes allow the molecules to move across the membrane by themselves. However, not all molecules fall in that category. There are molecules that are large and have charges, and cells need to be able to control where those go as well. So how can they do that? Well, one method that works well is a passive method known as facilitated diffusion. Now think about that word. To facilitate something means to help. So this is still diffusion, but it's diffusion with a helper. Let's take a look at it. Now, even though we're going to be using transport proteins as helpers in this method, it's important to note that facilitated diffusion is still passive. So we're still moving molecules from areas of high concentration to low concentration, and we're not using any extra ATP energy. It's just that the molecules are kind of awkward. They can't get across the membrane on their own, so they need a special helper to get them across. Facilitated diffusion uses helper molecules. And by helper molecules, I mean transport proteins. Those proteins that are embedded in the membrane and form tubes that form these little gates that let things pass through. One type of these little gates or transport proteins are known as channels. Channels are pretty simple tubes. Sometimes they can open and close when stimulated, but they essentially let molecules pass through their interiors, and they make a little gateway that allows things to pass through the membrane. Channels are often used for molecules that are a little bit big or also have charges. So for instance, here we have some chlorine ions, and because of that negative charge that that ion has, it's not going to be able to pass through this nonpolar interior of the membrane by itself. So it has to go through a gate. Now, most of these channels are very specific to particular types of molecules. So chlorine ions have their own channels, potassium ions have different channels, and so on. Another type of transport proteins are called carrier proteins. Now, whereas a channel protein acts like a big open door and allows molecules to just flow through, carrier proteins are more like a revolving door. The molecule kind of pushes on that protein and it changes shape and takes it in, and then it changes its shape again and kind of pushes it through to the other side. Whew, you made it. This is a hefty chapter. We've got one more part to do, but you are now finished with part 1. I would like you to go to your pre-lab for Lab 5. I'd like you to go to the top of page 3 now and see if you can complete that. And again, Bring all of these completed year turns to class with you so that we can go over them and confirm them. Great job. See you in class, guys.